Hi students, welcome to topic 3. This is on microbial culture. Topic 3 consists of a total of 5 parts. So this will be the first part. I'm showing the intended learning outcomes for topic 3. Here we would like to explain the purpose for the cultivation of microorganisms in the laboratory. Also, we need to know the importance of various chemical elements which are required by our organisms. We will try to describe the different groups of microbes according to their different nutritional requirements. You will need to know what's the difference between a culture media, as well as what's a pure culture and a mixed culture. Alright. Often we will also need to list the type of microbiology media based on their physical state, composition, and function. Okay, and you also will need to explain the use of appropriate microbiology media for various purposes. And finally, to describe various methods to maintain or to preserve microorganisms. So in this topic, just want to mention why do we want to culture microorganisms. Okay, first of all, to culture microorganisms, we need to grow them in sufficient quantities so that we can observe for their colony shape or size, basically their characteristics. So this will allow scientists or even the medical laboratory scientists to actually identify the species that's present in the specimen or the sample, allowing the differentiation between the different species and then characterizing them in the process. So when we have grown sufficient quantities of them for observation, okay, typically we will subject them for testing. Okay, in the field of medical diagnostic testing purposes, okay, we will subject them to antimicrobial susceptibility testing. Okay, so you can see on the image here on the right, okay, this is how we perform antimicrobial susceptibility testing. These white discs here are actually the antibiotic disc, right, that is placed on a lawn of microbes. This could be bacteria in nature. Not sure what's the species here, but it's often grown to a lawn here on a petri dish like this, on an agar plate, or depending on whether they are able to grow well on certain culture media, which we're going to see in the coming chapters, right? They were able to grow to a full lawn like this, and then you're able to determine whether the antibiotics are effective, whether the microbes are susceptible to it, or are they resistant to the antibiotics, just by observing how well there's a zone of clearance around the antibiotic this here. So in the laboratory session, I think it's the last session, you will have an opportunity to work on this in a dry light setting, right? So ultimately, by doing this testing, we are able to help the physician to guide patient treatment, especially for patient who is diagnosed with sepsis, bacterial infection. All right. In addition to medical diagnostic testing purposes, all right, we also want to identify and confirm species. So often we will subject the grown culture to biochemical testing, and this can be in the case of 16S ribosomes protein testing to confirm species of the culture. So in the current industry now, the multi-TOF biochemical testing is being done. MALDI stands for matrix assisted laser desorption ionization. TOF stands for time of flight. So basically, this is an instrument that helps to bombard the cultures if they have been spotted on a metal plate here, all right, and then determining what is the proteins carried, the ribosomal proteins to be specific, okay, carried by the bacteria, so that we can confirm the species. So once you have grown sufficient quantities of the microbes, okay, we will also often subject them for large-scale production. So in the industries of pharmaceutical or even biotechnology production, all right, here we're looking at virus propagation. All right, we grow sufficient number of virus to infect cells, and then we can produce vaccines from there. All right, antibodies perhaps. Okay, we're also looking at large volumes of bacteria. Here, these are we're talking about 20 liters, 50 liters or more in specialized containers are known as bioreactors, so that we can have sufficient quantities to have our enzyme, probiotic, or even antibiotic productions in large quantities here. So you can imagine that all these large quantities of microbes being cultured will enable us to produce those desired products here. All right. Another example is to produce food. Right. We typically consume food such as mushroom. These are fungi also in nature. Kelp, they are algae in nature as well. some of the major groups of microbes that we have come to learn about in this subject. All right, and finally, once we have cultured certain microbes, we also want to detect whether these microbes are contaminating in our environment. And often, you need to characterize them, differentiate them before you can actually make any conclusions out of this. So let's talk about microbial culture. What's the nutritional requirements for microbes? So nutrients needs to be provided to the microbes in a culture to grow them, right? So when we talk about essential nutrients are defined as any substances, be it elemental or molecular, that's required for growth, but cannot be synthesized by the organism itself, and thus we must provide it in the culture media itself. Generally, there is two types of nutrients, macronutrients, okay, and as well as micronutrients. So when we talk about micronutrients, right, it all depends on the quantity that's required by the microorganisms. In macronutrients, they require in fairly large quantities. So these are in grams per liter. Right. Some of the elements we are looking at here are your essentials, macronutrients, carbon, your hydrogen, your oxygen, right, your nitrogen, your phosphorus or phosphate, okay, or sulfur. Okay, in short, you might want to remember this as CHOMS, as a mnemonic for you to remember easily. Yeah, CHOMPS, CHOMS. Okay, as well as other elements, sodium, potassium, calcium, magnesium, iron, so on and so forth. So all these macronutrients are important to play important roles in cell structure and cell metabolism. Okay, micronutrients, MICRO, smaller, require much more in minute quantities. So these could be in milligrams or lesser. Some of these micronutrients includes your trace elements, such as your manganese, your zinc, your cobalt, right? Those are much more trace elements, and they play important roles in the microorganism enzyme function, as well as the maintenance of protein structure. So apart from the macro and micronutrients, referring to the nutrients based on the quantity needed by the microorganism, you can also classify their nutritional requirements based on their organic content. So organic nutrients such as your growth factors, your protein, your carbohydrate, lipids, so on and so forth, these are organic carbon sources that microbes often need in order to survive and to propagate. In addition to organic nutrients, you also have your inorganic nutrients, such as your mineral salts. So when we talk about organic nutrients, just remember they're often referring to their carbon content, those carbon-based contents that are outlined here, having the carbon molecules. Um, in contrast, the inorganic nutrients, those are often an atom or a simple molecule that contains a combination of other atoms other than carbon and hydrogen, which you find in the organic nutrients themselves. So when it comes to nutritional requirements, we know that water is the most abundant component in the cell, forming about 70%. So when you essentially remove all the water from the cell content, the dry weight of the cell remains, and this accounts for about 96%, consisting of the six elements of the carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfate. So after water, protein is in fact the next most abundant form based on this. So I want to impress upon you, all living things, including microorganisms, they require a carbon source as well as an energy source. Depending on what type of their carbon and energy source, we use different terms to describe these organisms. All right, let's look at some of the different terms that we use to describe this organism based on their carbon source. So now let's look at the different groups of microbes based on their nutritional requirements. So we can group microbes according to their nutritional requirements if based on their carbon sources. So for heterotrophs are defined as organisms that use reduce preformed organic substances such as glucose. And we know glucose is a good carbon source also. So heterotrophs are any organisms that obtain its carbon in an organic form. So these examples are some bacteria, okay, except for photosynthetic bacteria. Most of them, they actually are heterotrophs. They use glucose as a carbon source. Right, some species of protozoa as well as fungi. Yeah, right. Need to note that the heterotrophs are also dependent on other life forms. Hetero coming from a Greek prefix meaning others. So they are depending on other living organisms for food. Okay, so among the others organic molecules that we talk about, so proteins, carbohydrate, lipids, right, nucleic acid, these are organic nutrients that are available to the heterotrophs for them to absorb, right, in the simplest form ever. Let's for example, glucose, those are monosaccharides, all right, amino acids that are made out of your nucleic acids. Okay, the larger molecules, they need to be processed by the cell before they can absorb in their metabolism. Right, so some heterotrophs, they have different capacity, all right, to use different carbon sources. Some they only restrict to a few carbon sources as substrate, like for example some pseudomonas species. Okay, in terms of autotrophs, right, autotrophs are self-feeding, right, they use inorganic carbon dioxide as their source of carbon. Okay, so inorganic carbon dioxide is the source of carbon. So examples of this are your photosynthetic bacteria, your algae. All right, they have the capacity to convert carbon dioxide into organic compounds. Okay, and they are not dependent nutritionally on other living organisms. So autotrophs are actually self-feeders. Okay, they don't feed on themselves. Okay, but they are an independent organism here. So we can also group microbes according to their nutritional requirement based on their energy source. So when we talk about phototrophs, right? Phototrophs are organisms that use light as their energy source. So light in this case will be sunlight, all right? You also have chemotrophs. Okay, chemotrophs are microbes that obtain energy from the oxidation of organic or inorganic compounds here. So in the earlier slides, we have seen how we can classify different groups of microbes based on their nutritional requirements. Yeah. So when we talk about heterotrophs, we are looking at organisms that are dependent on others for food. Okay, and they utilize glucose, for example, as their carbon source. Okay, for auxotrophs, they are self-feeders. Okay, they are able to convert carbon dioxide into food for themselves. They don't depend on others. Right, that's all based on their carbon sources. We can also classify them based on their energy source. We saw what phototroph does. We also see what chemotroph does as well. All right. So for the sake of convenience, often we will actually combine the terms for their utilization of the energy sources. All right. In this case, the carbon and the energy sources together in a single word. Okay. For the sake of convenience, so like for example, for auxotrophs, we're looking at photoautotropes. So telling you that they are utilizing sunlight, for example, okay, as their energy source. Okay, so for example, these are examples of these are your synthetic algae species. All right, and this could be algae, can be plants, could be cyanobacteria as well. Okay, so the carbon source for them is still carbon dioxide, but the energy source for these photoautotropes are from the sunlight itself. Right, for under the category of autotrophs, you also have chemoorganic autotrophs. So chemoorganic autotrophs, for example, some of the metanogens species, they depend on organic compounds for the energy source, right? These metanogens species, they convert methane, CH4, to hydrogen and carbon, right? The carbon dioxide. So for carbon sources, it's still carbon dioxide for them. Okay, so the chemoorganoautotrophs, so they are quite dependent on themselves. So once again, they are using the organic compounds for energy and the inorganic compounds such as carbon dioxide as their carbon source. Right. Another category under autotrophs, we can classify, they are also known as chemolithoautotrophs. So these are uh, organisms which require neither all right, sunlight or um, organic nutrients. They rely totally on inorganic minerals itself. So some of these examples are the deep sea mineral bacteria, which often they call it rock eating. So in this category of organisms, they are utilizing the inorganic compounds, the minerals, for example, found in the sea, right, as the energy source, and the carbon source is still carbon dioxide. For heterotrophs, right? Heterotrophs, we can classify them as photoheterotrophs. Right? Again, these are utilizing sunlight as their energy source. These are microbes or organisms, and often you can find these like example, the purple and the green photosynthetic bacteria. The carbon sources are still organic in nature. They still require the organic as the carbon source, organic compounds as their carbon source. We have another category also known as chemoheterotrophs, right? So again, heterotrophs, they're dependent on others for food. In this case, they will depend on others for nutrients in terms of the organic compounds. So here we're looking at protozoa and fungi. After learning the different classification of the different groups of microbes based on their nutritional requirements, how will you classify or describe these microbes? We know that there are different groups of microbes. For the bacteria themselves, right, most of the bacteria are actually chemoheterotrophs. Right, chemoheterotrophs, again, they get their carbon and energy sources from organic compounds, right? Often they process these organic molecules by respiration or by fermentation, and then this releases ATP, adenosine triphosphate, which is a form of energy, right? Chemoheterotrophs like them, they also use aerobic respiration, which is a primary source of energy healing pathway. So some of this includes the fungi as well the algae, right? They are also using the aerobic respiration. So the fungi, they are chemoheterotrophs as well. The algae, majority of them, okay, they are actually photoautotrophs, using sunlight as their energy source, as well as carbon dioxide as their carbon source, right? They are self-feeders, they don't depend on others for food. And finally, for protozoa, often you see that majority of them, they are actually chemo heterotrophs. So I want to bring in two different terms where you can classify microbes based on their nutritional requirements. So we know that not all microbes require all growth factors. Remember, growth factor is an organic compound such as amino acid, a nitrous base, or vitamin that cannot be synthesized by an organism and must be provided as a nutrient in the nutrient culture media. So some of the microorganisms, they can synthesize all the growth factors they require. These microorganisms or microbes are called prototrophs. Some organisms are like this. Other organisms require certain growth factors to be included in the growth media or in the culture media themselves. So we call these microbes as auxotrophs. So like for example, you have histidine auxotrophs, right? Microorganisms that cannot make the amino acid histidine. So you will need to include histidine as the amino acid in the growth media itself. Okay, the auxotrophs are microorganisms themselves. They are often genetic mutants. So, i.e., they lose their ability to synthesize certain essential nutrients. And we know these are essential nutrients such as your amino acid or your vitamins. So in summary, we have covered different groups of microbes that we can classify them based on their category. So for auxotrophs, where you have phototrophs that depends on sunlight for the energy source, carbon source in the form of a carbon dioxide to convert to food. All right, these are some of the photosynthetic organisms that we come to know about, right? So you also have chemo auxotrophs. Those are dependent on organic compounds as the energy source, the carbon dioxide as the carbon source, and these are some of the methanogen species that we come to observe. All right. Under auxotroph category, we also have chemoorganic auxotropes. Chemoorganic autotrophs. So these are microorganisms or organisms that utilizes inorganic compounds such as minerals and as their energy source, as well as carbon dioxide as their carbon source. Right. In contrast, heterotrophs, right, which utilizes organic compounds and depend on others for food, they can be classified under photoheterotrophs using sunlight. Right, carbon source being organic compounds. These are some of the purple and green photosynthetic bacteria. Right, you've got chemoheterotrophs. These rely on organic compounds also and the conversion of those organic nutrients from other organisms. So this includes the fungi, the protozoa, and many other bacteria. All right. The two remaining categories here are saprobe and parasites. They are for the former, it usually metabolize the organic compounds or matter of dead organisms. Again, organic compounds are their main carbon source once they are broken down, and often these are fungi or even bacteria as decomposers. The last category is heterotrophs, the parasite category. They actually depend on living hosts, all right, and they utilize the tissues, the fluids, as parasites. So some of them may be pathogen as well for their organic compounds as a carbon source. So in summary, I have touched about why we are culturing microorganisms for various purposes. The main aim is to actually grow sufficient quantity so that we can observe them and utilize them for the various purposes that we talked about earlier. Right? There are also essential nutritional requirements of a microbial culture. We have broken down into the macronutrients. These are your CHOMs, as well as your other sodium, potassium, calcium, and other metabolites here. All right, we also look at micronutrients. These are much more trace elements like your manganese, your zinc, and your cobalt. Right. We are able to classify different groups of microbes based on a nutritional requirement as well. We talk about heterotrophs and also autotrophs, and then the different classification based on their carbon and energy sources, where we combine both of their sources together, so much so that we can identify if there's a microorganism that uses sunlight, for example, right? Those are usually photoautotrophs, and they are dependent on their cell feeders. All right, so this concludes the first part of topic 3. I will see you in the next part. Hi students, welcome to part 2 of topic 3. Topic 3 is focused on microbial culture. Here I'm showing the learning outcomes for topic 3. So in part 2 of this e-recording, we're going to look at defining culture and culture media, right? Describing mixed and pure culture. We'll look at some of the aseptic techniques, as well as describing some of the methods that we can do sterilization of the media and the equipment here in the laboratory. So when we talk about microbial culture, often a culture is a collection of microbes, right? The microorganism itself. So we remember in the previous part of this e-lecture, we are talking about growing sufficient amount or quantity of these microbes to study them, as well as to use them for industrial purposes, right? So microorganisms can be cultivated or even grown in laboratory. And every batch or sample that we grow, we call it a culture, right? A culture indeed is a collection of microorganisms, right? So to culture can also refer to the act of making a bacterial culture itself. As the bacteria grow or the microbes grow, they quickly use up nutrients present in the culture media, right? To make a culture or to grow the culture, here's what we define it as, right? And once they use up all the nutrients in the culture media, they also at the same time produce waste, and these waste can be very toxic for their own growth if they're allowed to accumulate to a high amount, right? So to prevent that, and we want continuous growth of our microbes of interest, we will actually transfer a small quantity of these microbes, right, into a fresh preparation or culture media. So from a pre-existing culture to a fresh culture, we call this a subculture. Okay, and when you do a subculture, often we will get a sample of it. The sample, we call it inoculum, and then when we do the transfer over to a fresh media, we call it the inoculation, right? So this will serve to give fresh nutrients in the fresh media to the microbes, all right, as well as diluting the waste that's present in the previous culture itself. So remember, to subculture is to make a culture from a pre-existing culture. right? So this whole process is a subculture. Right, the subculture is defined as a new culture again, and moving forward, you will be allowed to grow, and the whole process starts again. So let's look at the difference between pure and mixed culture, right? So in a mixed culture, culture that usually contains more than one species of microbe, this is called a mixed culture. So if you see on the schematic here on the left hand side, a mixed culture often contains many different type of species of microbes, and hence different types of colony that you can expect. Different microbes, different species have different colony shape and sizes, right? As shown here, right? That's grown on the agar plate. Okay. In contrast, for pure cultures, often these are only single type of species or microorganisms, and often they are of a similar characteristic, i.e., these could be gray-looking or flattish-looking colonies on the agar plate itself, right? So colonies, I just want to mention here, they are often cultures that are visible on the surface of the solid media like agar plates like this, right? Different species of bacteria and even fungi can have different morphology on the colony, right? Different color, different shape, different sizes, as I mentioned. Texture could be different also, right? So these are the characteristics that we can study, and it will help us to differentiate between the different types of microbes, right? Macroscopically, so using your naked eye, right? When you do different stainings that we're going to talk about later on, then you are able to differentiate them. So all in all, just need to know the difference between the mixed and the pure culture. So we briefly touched about culture media just now. So culture media here we define as a mixture of different compounds that provide all the nutrients that are required for microbial growth. So if you remember in part one, we talk about auxotropes, right? Auxotrope lacks certain capability to synthesize certain essential nutrients, like for example histidine as an amino acid. So in order to grow them, you need to add histidine into the growth culture itself. Right? The culture media is often also known as the growth media. So when you grow microbes, often we will call them as microbiological media as well. So all the same. So in terms of microbial culture media, there are generally two types. The first type will be the liquid form, right? These are often we call a broth, right? A liquid form of culture media. The second type is a solid form, an agar, which you have seen earlier. These are solid form of the culture media, and they usually come with agar. And agar, as you know, it is a very solid media that people come to associate with desserts as well, right? But in this case here, we're looking at microbiological growth or culture. So the choice of this type of media depend on how we want to culture and the type of microbe that you're culturing, right? Some microbes prefer to be cultured only on one type of media, right? Many agar, many other protozoa, and some bacteria can also be cultured in broth. So it depends on the species and the type of microbes that you are using, all right? And then what are you using the culture for, the purpose of it. So here I've just include some representative images here. On the left here, this is how a liquid broth will look like. Typically we put it in tubes, Falcon tubes in 15 mils or 50 mils, 50, right? The solid agar culture media often in petri dishes like this, right? Depending on the agar and depending on the recipe of the culture media, they may look colorless to yellowish, and some are red, as you will see later on with the different types of media for microbial growth. I want to touch on aseptic techniques. You will come to learn about this in more details during the practical, but aseptic techniques is defined as a set of specific practices and procedure performed under controlled environment so that there's a minimum contamination by undesired organisms. So we want to minimize the risk of contamination by undesirable organism apart from our microorganism of interest. So often we will need to protect our sample, our culture, and all other media and equipment from contamination by surrounding microbes. We know microbes are in the air. In fact, the air that we breathe in may contains fungi spores as well. Our hands also are covered with microbes, our benches, your tables, and so on and so forth. So aseptic techniques are essential, right? And you will get to practice this during the practical. Importantly, it also helps to protect the user from potential harmful microorganisms. I want to switch gear now and talk about the methods for sterilization of culture media and laboratory equipment. right? So why the need to sterilize media and the equipment associated with it, right? We want to do this because the aim is to destroy any microbes that's present on them before we use it, so that we can ensure that only the intended microorganism is cultured in the process. We won't inadvertently introduce other undesirable contamination or other microbes into our microbial of interest. So in the laboratory, sterilization can be achieved through different means. It can be done through autoclaving, heating in the hot air oven, flame sterilization, as well as filter sterilization. So we will look at all this in more detail in the coming slide. So just to re-emphasize again, sterilization is a process that results in a condition that is totally free of living organism and other living things, right? It is essential to remove unwanted microorganisms from our culture media, instrument, container before using them. Okay. So now let's take a look at the methods for sterilization. Autoclaving is one of the most common method used in the laboratory, especially in the microbiology lab. So here we are subjecting your materials, your media into with steam under pressure, also known as steam sterilization. Steam, we know, under pressure can achieve an effective kill. The autoclave machine, as you see on the right hand side here, is used to provide a high temperature steam under pressure. So we are looking at the common settings of 121 degrees Celsius at 15 psi for 15 minutes. Okay, or in another setting of 115 degrees Celsius for 10 psi pressure for 10 minutes. So these programs are important because these will allow you to kill vegetative cells as well as endospores. Okay, there's a need to monitor the effectiveness of this autoclave. Often we will do this by mechanical monitoring, so looking at the temperature readout. Do we achieve the desired temperature as we have set out to do? All right, the time, is it a complete cycle for 15 minutes or 10 minutes, as well as the pressure? Do we reach the certain pressure that we have outlined here to do autoclaving? So this will be monitored as we do this to ensure that there's complete sterilization. Right, chemical monitoring, we can do this also by using heat sensitive tape or even color strips to tell you that sterilization or even autoclave has been completed, as well as biological indicators. There are indicators that are sold outside that contains microorganisms, and they have color changes in them when you have effectively killed the microorganisms with autoclaving. So just to note that the items that are sent for autoclaving here, they must be thermostable and not sensitive to moisture. So here we're looking at a lot of steam, pressure, high temperature. So they need to be thermostable. So you can't use, like for example, a media that has antibiotics in there in autoclave. The high temperature will kill off any of the antibiotics present in there. Okay, apart from autoclave, we also can perform heating in a hot air oven. So here, materials and the equipment that may be sensitive to moisture, and you cannot use it for autoclaving. Yes, you can heat it in the hot air oven. So this will be carried out up to 250 degrees Celsius for several hours, and aims to kill the vegetative cells and the endospores, depending on the temperature and the duration that you have set in the program. So once again, these are suitable for materials and equipment that may be sensitive to moisture, right? Because here we're not dealing with steam, here is only hot air heating. Another method for sterilization of our culture media is through flame sterilization, and this you will be doing hands-on in our practical sessions. So flame sterilization, often these are for non-flammable equipment that are used over and over again, so very reusable within a short period of time. So here we're looking at your inoculation loop and your wire, right? You have been asked in the laboratory during your practical to actually heat your wire and your loop to red hot or orange, so that you can effectively sterilize the loop itself before you take an aliquot of the microorganisms, be it in a liquid broth or in a solid media. So often we also have other equipment that can be subjected to flame sterilization. So this includes your glass spreader, often used for a spread plate, all right, your scissors, your spatula. All this can be sterilized with flame. So some laboratory, they will actually perform surface sterilization using alcohol and flame. all right, apart from using flame alone. Okay. So another method for sterilization is through filter sterilization, also known as filtration. So these are suitable for fluids and solutions that are not thermostable. So you can't use, like for example, antibiotics. You place it or add it inside a liquid media, a nutrient agar media, and then subjecting it to autoclave. This will deactivate or even destroy the antibiotics. And hence they are, because antibiotics, we know, they are not thermostable. They are highly heat sensitive. So filtration can be done through membrane filtrations. So once again, membrane filtrations involving or in fact should be used for fluids and solutions that are not thermostable. All right. Based on size exclusion, right? These are filter membranes that have different sizes. They can be in 0.45 micron. They can be in even smaller filter size, up down to 0.2 micrometers, right? So based on the size itself, you will have effectively trapped the different microbe cell on the membrane because they are too large to pass through the different pores, right? Generally, we will use 0.2 micron to 0.45 micrometers filter membrane to filter our solutions or our fluids, especially they are not thermostable, right? So liquid and gases that cannot be sterilized by heat, right? They can be filtered sterilized through the membrane, as the small pores itself will allow the liquid itself to pass through, but not the microorganism. So just a note, after filtration, whatever that is being filtered, right, the downstream product, that is called a filtrate, and that filtrate is therefore sterile. Okay, so as I mentioned just now, these are the common membrane pore size that you can come to read about and encounter them in the laboratory. Okay, 0.45 micrometer, good for removing bacteria and larger cells. Go lower than that, 0.22, the pores are even smaller. That in fact effectively good for removing large viruses itself. So I included an image here that shows a membrane filtration unit. This unit itself, if you place your liquid here and then you pass it through this filter membrane, you can collect your filtrate here, which is sterile. So this whole filtration unit is actually driven by a vacuum pump, so that your solutions that's added here will pass through the membrane filter to give you your desired filtrate. So we have looked at what is culture. Culture is a collection of microorganisms. We also look at what is culture media. Culture media, it contains all the fresh nutrients essential for the microbes to grow in. We also talk about subculture. Subculture involves taking an inoculum or aliquot of your microbes from a pre-existing culture and moving it to a fresh culture containing media. So in this case here, your aim is to actually continue to propagate the culture that you intended to, and then this will provide new nutrients for the microbes as well as diluting the metabolic waste that they have accumulated. We also talk about pure and mixed cultures. So pure cultures are basically there's usually a single species in your culture media, whereas the mixed culture are usually containing more than one species in the culture media. Right. We also talk about two different types of culture media. They come in two flavors. Two of them. One of them is the liquid broth or a solid, usually in the form of agar. Okay, and we also talk about different sterilization technique with the aim to actually remove and disinfect any contamination. So this includes the most commonly used autoclave, as well as heating in the hot air oven, using filter sterilization, and the use of flame for flame sterilization. Right, so this concludes the end of part 2 of topic 3. I'll see you in the next part. Thank you. Hi students, welcome to part 3 of topic 3. Topic 3 is focused on microbial culture. Okay, so let's get started on part 3. So in part 3 of topic 3, we will be focused more on listing the type of microbiological media based on the physical state as well as composition. In part 4 and 5 of topic 3 in this e-lecture series, we will talk more about their function as well as their appropriate use for various purposes of this microbiological media. And to cap it off for this topic, we will be talking on some of the methods that we can use to maintain and preserve microorganisms. So let's jump into this. So in the earlier parts of this video, we have actually defined what is a culture media. Culture media can be a mixture of different components that provide the nutrients required for microbial growth. It contains all the nutrients required for its growth, and in order to grow well, it's important that the following media, such as water, carbon salts, nitrogen salts, inorganic salts, growth factors, and hydrogen ions are all present. So when we talk about culture media, we also talk about it as a growth media itself. It's the same as well as microbiological media. And generally it comes in two types. Liquid, it can come in a broth form, or solid, and usually in the agar form. So with the microbiological media, we can categorize or classify them based on three different parameters here. Based on their physical state, based on their composition, as well as their function. So let's look at the physical state of this culture media intended for microbial growth. First, for liquid media, usually these are in the case of nutrient broth, right? As well we mentioned earlier. For solid media, this could be in the form of nutrient agar, right? So all this media, it contains the necessary minerals, vitamins, growth factors, allowing the microbes to grow. In the solid media, usually agar, which is one of the components, is added up to 2% volume per volume as a solidifying agent. This is used, especially some of the microorganisms, except for some marine type of bacteria, they can catabolize agar as one of their source of carbon. Right? Agar solidifies at roughly 45 degrees Celsius. So at this temperature, you can actually add heat-labile substances such as antibiotics into the molten agar itself, and it will still be fine. So when we talk about heat label, we're talking about heat-sensitive substances such as antibiotics. Okay, apart from liquid solid media, I just want to introduce a third media, a semi-solid media. Okay, for example, in the case of this mortality media called the sulfur iodide. All right, this is usually used as a media to study microorganism mortality. All right, the media agar as a component added is much more lesser at 1% as a solidifying agent. So this will allow scientists to look at how microbes move in terms of their mortality using this semi-solid agar. When it comes to solid agar, microbes often grows at colonies on the surface of the agar itself. So you can see on the right here with this schematic, they have a colony that comprises of millions of cells. You also have some of the agar plates that can grow very well with fungi as well. Alright, so solid agar, often in the agar form, solid cultures, they are very useful for isolation to give you pure cultures. Remember, pure cultures are only cultures that contain a single species. Easier to detect for contamination. All right, if you have a different color popping up from your petri dishes of your agar, you will know that they are not pure anymore. Right. However, solid agars are very tedious to prepare, especially in large quantities. All right, and they often give you small yields of your intended microbes also, and it's more difficult to harvest them. So in contrast to solid culture, here we're looking at liquid culture here, or a broth culture. So often when you have growth inside a broth, it will often appear as turbid. So something like this will become very turbid because non-inoculated broth are usually clear. You can easily prepare large quantities of the media, and large quantities of the microbial cells can also be cultured also. In the liquid broth, they are easily harvested and diluted as well. However, because of its nature, it's difficult to tell whether is it contaminated with unintended microorganisms. And once it's contaminated, it's difficult to salvage already. So in terms of the chemical composition of the microbiological media, the type of media itself depends on various. You have two different types of media. First, we have the defined or the synthetic media. They are made from known components. So, i.e., we know the exact composition in this defined media, and hence the name. Very easily duplicable. You can easily replicate them. So when we know that they have the exact composition, I'm talking about the exact quantity of chromes, sodium, potassium, and so on and so forth. They are known. So remember chromes from the earlier topic, we are looking at the carbon source, the hydrogen, the oxygen, nitrogen, phosphorus, and sulfate. All right. For undefined or complex media, usually they contain natural products. All right, the exact quantity of chromes and the rest of the ions here are unknown. All right, and it differs from batch to batch also. All right, exact composition we don't know. It's difficult to duplicate exactly as well because of the batch to batch variation. Okay, in both the media and the culture. Right, so when we say again chemically defined or synthetic media, we know the exact amount and the nature of each of the individual ingredients. In the undefined or complex media, the exact composition of some of the ingredients are unknown. Right, so defined media, we know that they are made from known components. All right, and exact composition is known, and hence we can reproduce that. But undefined media, also known as complex media, we don't know the exact composition and we cannot completely replicate or duplicate it. Okay, the complex media are more commonly used for the routine culture of the microorganisms, since they are able to meet the nutritional requirements of many of the microorganisms. This is for the complex, or also called the undefined media. So here the table shows one of the representative recipe for one example of the defined media. So you can see that it contains inorganic salts as well as the source of the carbon. So in terms of sodium bicarb, so this is NaHCO3. All right, bicarb is available as a soluble form of carbon dioxide for carbon fixation by the bacteria. Okay, especially for bacteria like who are autotrophs, right? They can use this as their carbon source. All right, we also has sodium diphosphates. Okay, this provides the phosphorus groups or the phosphorus ions buffer against any of the change in the media, changes in terms of the pH level. So once again, defined media, we know the exact composition because in the amount, in terms of grams per liter, we know the exact composition in the media itself. Okay, for undefined media, right, this is a nutrient broth recipe, a representative one. Okay, often the exact composition of the individual components here we won't know. Although we say that we can add a certain amount grams per liter in terms of its concentration, but the exact component of its natural products here, so such as the beef extracts, right? Beef extract contains heaps of other molecules in there. This could actually provide you with a carbon, nitrogen, sulfur, and so on and so forth, the different growth factors. We will not be able to know the exact quantity of them. Okay, same for pepton. All right, pepton also contains, these are natural products that contains different sources of carbon and also macronutrients essential for the microbial growth. So when we talk about natural products in undefined media, these natural products are often water-soluble extracts. So like for example, yeast extract, beef extract, so on and so forth. Right? It provides minerals, vitamins, the organic growth factors, organic carbon, nitrogen, and other of the essential nutrients for the microbial growth in the culture media. Natural products themselves can also be digests of proteins. So these might be, for example, casein, hydrolysate, peptones, tryptophan. These provide essential amino acids as well as the organic carbon, nitrogen, and sulfur. Once you have a protein that's been digested, these are actually forming your amino acids, which are the building blocks of your proteins. So digest of proteins found inside natural products, we will be able to know its constituents, but we will not know its exact compositions. So apart from the proteins themselves, the digest of proteins, you have other components that can be present inside the natural product. So this could be blood or serum that provides complex growth factors for very fastidious microbes, right? When we say fastidious, we are talking about microbes that need special requirements, right, or a special environmental growth condition, right, in order to grow well. So some of these include the Listeria species, which needed blood to grow well in their natural state. So in summary, in part 3 of this video, okay, we have listed some of the microbiological media based on the physical state as well as composition. We talk about the different cultures, physical state. It could be a solid state. Often these are in the form of agars. All right, liquid cultures can be in form of broths or semi-solid cultures, where we see some of the sulfur iodide mortality media that scientists use to study mortality the movement of the microbes themselves. We also look at microbiological media based on their compositions. All right, we have defined, also known as synthetic media, where the exact composition is known. Okay, we can reproduce the recipe easily, but we also have complex or undefined media, where often we wouldn't know the exact composition, but it's usually this media that supports a wide range of microorganisms. All right, so in the next part of this videos, we will be focusing on the functional types of this microbiological media. So see you then. Hi students, welcome to the last part of topic 3. Topic 3 is focused on microbial culture. So in this last part of the topic 3, we'll be focusing on the microbiological media based on their function. In the earlier part, we have talked about the physical state as well as its composition and the different types of microbiological media. We will also be explaining the use of it as well as the different methods that we can maintain or preserve microorganisms. So in terms of the function of the types of microbiological media, the functional types of microbiological media can be classified based on the purpose of their use. So generally, we have the general media, the minimal media, the selective media, the differential media, the enrichment media, as well as the specialized media. So media categorized again based on the purpose of their growth. So you need to know these 6 functional types of media. Let's dive into each of these media as we talk about its function in the isolation of microorganisms. So let's look at general media. General media are actually complex media. They contain natural products. Recall from the last part of our video, we talk about natural products having a good source of carbon, nitrogen, phosphorus, sulfur, as well as other minerals, as well as growth factors. They are also good energy source. Most of the non-fastidious microbes will be able to grow on such media. Hence, the general media itself allows the growth of a wide spectrum of microorganisms. All right, so they have a ability to support growth of a wide spectrum of wide variety of microorganisms, except the fastidious ones. Remember the fastidious one, they need special growth factors. They need special condition to grow. Like for example, special components of it could be blood, could be serum. Then that will allow them to grow well. Because the fastidious ones, they are nutritionally demanding. So some of these general media, for example, are the nutrient broth agar or nutrient broth itself if it's a liquid form. All right, or the tryptone soya broth or agar. These are general media that are able to support the growth of a wide variety of microorganisms. Here I'm showing a recipe of a general media known as tryptic soy broth. Tryptic soy broth is an example of an undefined or also known as a complex media because it contains the following ingredients here: tryptone, peptic digest of soya bean, and other constituent including glucose and water. So the peptic digest of protein of this protein soya bean actually provides a high carbohydrate and vitamin content, and hence making it a undefined media. The two natural products here, as I mentioned here, this will be the peptide digest of the soya bean meal as well as the tryptone. These are the two natural products that will help to support the growth of a wide spectrum of microorganisms. Look at the minimum media. Minimum media are chemically defined, hence defined also known as synthetic media often. The defined media in minimum media, as the name suggests, they contain minimal nutrients to support the growth of specific strains or species of microorganisms. So more often these are mainly consist of minerals or even simple organic nutrients such as glucose only, just to provide the carbon and the energy source for the microbes. All right, it's useful for screening auxotrophic mutants. All right, remember mutants, auxotrophic microorganisms are mutants that are deficient for certain biomolecules such as amino acids. Okay, and it helps into investigate what's their minimum requirement, the bare minimum in order for them to grow and study them. All right, so one example of it is M9 salt agar that are useful for screening auxotropes. So note here that the minimum media usually contains a carbon source and other inorganic salts and water. No amino acids are actually provided here. Okay, this table shows the minimum media M9 media, okay, containing inorganic salts as well as just glucose as a source of carbon. So this provides the carbon and energy source for the microbes growing in the culture media, be it a liquid broth or solid culture. So let's look at selective media. Selective media are actually media that contains inhibitors that allow the growth of certain microorganisms but prevent the growth of others. All right, so selective media actually allows you to have rapid isolation as well as preliminary identification of the desired microbes. So it really suppresses the growth of undesirable microbes and favor the growth of a particular microorganism, depending on the media constituents themselves. So only microbes that are not inhibited by these constituents or substances within the media can grow. So like for example, in this image here that shows aspergillus. Aspergillus is a fungi. It grows really well on potato dextrose agar. So only aspergillus will be able to grow in this condition here because it contains substances that allow it to grow while inhibiting others from growing. All right, so another example here will be the eosine methylene agar, blue agar, or EMBA, eosine methylene blue agar. This is a selective media, but you may also consider this as a differential media, which we will talk about later. So mainly look at this as a selective media because this allows you to grow gram-negative bacteria and we are able to use it to differentiate between fecal and non-fecal bacteria. So typically fecal bacteria, gram-negative bacteria, they ferment lactose, and these are actually our bacteria that's found in our intestine. So upon fermentation of the lactose, and you may include it in the agar itself, they produce acid. In acidic conditions, it changes a pH substance in there that allows the colonies to produce a complex color of purple and metallic green. Right. So, but however, in contrast, non-fecal bacteria, they metabolically they ferment lactose much slower and they produce smaller amount of acid relative to the fecal bacteria. They can produce large amount of acid upon fermenting lactose as a carbon source. So in the end, the non-fecal bacteria, they produce small amount of acid and hence their colonies are of a different color. They may appear as brown or pink as what you see in the image here relative to microorganisms such as the gram-negative fecal bacteria that produces a dark purple or metallic green color. So in the previous slide, we mentioned about the potato extract agar. So that allows the fungus Aspergillus to really grow really well. So what this is in due to the fact that this potato extract agar, it has potato extract that has a high carbohydrate and low nitrogen content, which limits most of the growth of most bacteria. In this case here, it permits or even allow and favors the growth of your Aspergillus fungi. Right. In addition, we also have the addition of lactic acid here. Right. Lactic acid is actually added separately into this media so as to titrate the pH to a low 3.5. So only allowing certain microbes such as acidophiles to grow. Because don't worry about the term acidophiles here. These are mainly microbes that favors growth or grow well under low pH, acidic conditions, acidic pH. So we'll talk about this in topic 4 later. All right. So once again, because of its low pH as well as its high glucose concentration, all right, high carbohydrate content, it inhibits most of the growth of the bacteria, allowing and favoring the growth of fungi such as Aspergillus. So now let's take a look at differential media. Differential media contains components that interact in different ways with different microbial species. The differential media gives us the visual differentiation by appearance. So this can be in the form of color of the colonies and as well as the surrounding agar that the colonies are sitting in. Right, to distinguish colonies of one microorganism to another. So your differential media will allow the growth of all the multiple types of microbes, but it's the different color tones, perhaps the different morphology they exhibit in terms of color changes that will help you to distinguish different between the different species. So it usually involves nutritional or nutrients utilization of the microbes. The differential media itself takes the advantage to the fact that different bacteria utilize nutrients of any given different media in different ways. If acid or alkaline production is involved, often a pH indicator is always included, and this could be phenol rate or neutral rate. So in the case of McConkey agar, McConkey agar contains lactose as the carbon source, and the pH indicator that it uses is a neutral rate dye. So this McConkey agar media is able to differentiate lactose fermenters from lactose non-fermenters. Right, so colonies of bacteria that uses lactose will produce acid and they will become red in color. So this is shown here on the right hand side image here. The change in the acidity changes the color of the neutral red, right? And then they are got absorbed into the bacterial cell itself. In contrast, non-lactose fermenters are colorless here. All right, so they appear as colorless. So this allows you to differentiate the different species in terms of their nutrient utilization. In this case, this will be lactose. Apart from McConkey agar, blood agar are also differential media. We'll look at more in depth into blood agar in the next slide. I also want to introduce the differential media of the triple sugar iron agar. The triple sugar iron agar, also known as TSI, is a differential microbiological media also that help us to identify enteric bacteria based on their ability to ferment glucose, lactose, and sucrose, as well as to identify whether they produce gases or not, such as hydrogen sulfide. So triple sugar iron agar, TSI, the triple sugar already tells you already. It has three different types of sugar. So lactose here, okay, sucrose, as well as glucose. So it uses phenol rate in this case as a pH indicator. It will turn yellow for acid conditions, red, right, if it's an alkaline condition, as well as black, all right, to detect for hydrogen sulfide production. So in this case, the image here showing the slant agar, the triple sugar iron agar. If you have a yellow bottom here as well as a yellow slant, all right, and you can clearly see here there's some bubble formation. So this tells you that the microbes that are being inoculated here, it can utilize several of the carbohydrate sugars here, including lactose, and has produced gas. So it ferments lactose, possibly glucose, as well as sucrose, the three types of sugar in your triple sugar iron agar. All right. You also have the red slant here with a yellow bottom. So these are indicative of microbes that ferment glucose only, and this will show up in this kind of phenotype. You have the red slant and also the red bottom, so indicating there is no fermentation of sugar at all. It's just utilizing peptone as a source of nutrients here. All right, and then the blackish bottom here only indicates that the microbes inoculated here produce hydrogen sulfide. All right, so some of the microbes you can come to expect with such phenotype, like for example, in the case of tube A, tube A you have the yellow bottom and the yellow slant. Typically, these are your E. coli. Okay, they also produce gas, so they can utilize the three types of sugars here. All right, in tube B, all right, you have the red slant and the yellow bottom. These are typically Salmonella species. All right, for a blackish bottom here of the slant agar. All right, this could be some of the Shigella species that produces hydrogen sulfide. And finally, for the red slant and the red bottom, this could be some of the Pseudomonas species. So let's look at the blood agar. Blood agar is also a example of a differential media, right? Remember, differential media, they allow us to grow multiple types of microbes, but are designed in such a way that it can tell us visual differences among the different colonies. And the differentiation can show up as variation in the microbial colony size or in the color due to the media color change. All right. So in this case, this is a representative blood agar recipe. All right. It contains the peptone. It's a complex media with yeast extracts. It also has sheep blood in there. Okay, the blood is added, all right, before into the cooled molten agar before we pour it into the petri dishes. All right. In the case of this differential media of blood agar, all right, it allow us to identify different bacteria species that lyse blood cells differently. All right, so when we look at the Streptococcus species, the Streptococcus species have different variations, right? This variation is due to the fact that they contain hemolysins, right? So hemolysins are actually enzymes that break down red blood cells, right? So as to produce hemoglobin for the bacterial growth. All right, so some of these Streptococcus species, when they are grown on the blood agar, okay, they are classified into different types of hemolysin bacteria producing um, um, these enzymes. All right, so in this media here, as you can see, all right, if it's alpha hemolysin producing bacteria, all right, they are only able to um, have partial lysis of red blood cells, okay, resulting in a visual observation of a greenish halo color around the colony themselves, right? So in contrast, if you have streptococcus that are beta hemolysin, all right, so they have a complete lysis of red blood cells, so hence you can see that there's clearing around all the colonies here, all right. um, Whereas in contrast, bacteria which has no hemolysin enzyme as well, right? They are termed as gamma hemolysis. There is no lysis of red blood cells, those that are seen here on the right hand side here. So although blood agar, as we've seen earlier, it's a differential media, it's also a type of enriched media that we will see later to allow for the growth of fastidious microbes. So fastidious means they need certain constituents or nutrients in order to grow. These are very fussy microbes. Okay, but the point I'm bringing across is that a single media itself, not only it can be a differential in its entirety, not only it can be a selective media in its entirety, but also it can be both selective and differential as well. And this is in part due to its various ingredients or constituents within the media itself. So differential media and selective media have both differential and selective property. They're able to differentiate colonies of different uh, uh, species, all right, by displaying visual differences between them. The variation, again, as I mentioned, can be due to their color, can be due to their size, due to the media changes around them, all right, as well as some of the formation of gas bubbles or precipitate, which we will see in the triple sugar iron agar later. All right, but often they also exhibit selective properties also. So the selective properties allow them to favor certain growth, right? As we've seen in the potato dextrose agar, right? The selective properties itself, okay, it contains the constituents or substances that inhibits the growth of a certain microbes and promote the growth of the other, right? So often the differential and selective media, they are all used for presumptive identification of microbes, right? From food, clinical or environmental samples. Okay, some examples of this are McConkey agar as well as Meditor salt agar, right? Let's take a look at the McConkey agar recipe. You can see in the McConkey agar recipe, they have the presence of bile salts and crystal violet, as well as a pH indicator called neutral red. All right, so it has a carbon source also, which is your lactose. So this media is basically used for the detection of coliforms from the environmental or food samples. So remember, coliforms are bacteria that's found in the fecal matter of all animals and human, warm-blooded. The coliform bacteria themselves are lactose fermenters, so they will utilize this, the lactose here. So the bile salts and the crystal violet largely inhibit gram-negative, or sorry, gram-positive bacteria. So the bile salts and crystal violet will inhibit gram-positive bacteria, and hence you will select for gram-negative bacteria here. All right. So fermentation of the lactose will produce the acid by these gram-negative bacteria. So producing acids which lowers the pH. So in the presence of neutral rate, which is the pH indicator, this change the color of the colonies to red or pink. So indicating these are all the lactose fermenters. All right, so this allows us to differentiate between the lactose fermenting coliforms, which are often gram-negative in nature, from the non-lactose fermenting coliforms. So on the right here, you can see here they appear as red or pink colonies. Those are the lactose fermenters. These are your coliforms that are gram-negative, okay, as well as your non-lactose fermenters, which are colorless in nature. So essentially, this is how a differential media will work. Okay, another differential and selective media is also known as the medital salt agar. So the medital salt agar, as you can see, it contains medital, all right, as well as phenol rate. Phenol here is the pH indicator. So this media is used for the differentiation of pathogenic staphylococcus from other staphylococcus samples. This high 7.5% salt actually inhibits most of the bacteria, okay, appearing or in fact only allowing salt-tolerant bacteria to grow. So typically for S. aureus, all right, Staphylococcus aureus, they ferment medital and it will appear as yellow color colonies here, as you can see on this top right image here of the agar plate. All right, in contrast here, different species, all right, they will exhibit different color because they're non-medital fermenting. Like for example, in this case here, this is the Staphylococcus epidermis. All right, so there's no change in color at all for Staphylococcus epidermis. So we'll reach specialized media. So specialized media are actually media that's used for purposes other than growing, isolating, or even differentiating microorganisms. So like for example, we have the transport media. They are often used to maintain or preserve specimens for transport, held for a period of time during the transportation stage, before any analysis or even we use them under stable conditions. We also have reducing media. All right, reducing media is special because this reducing media contains substances such as cysteine that can absorb oxygen and slow down the penetration of oxygen in the media. They're often used to grow anaerobic microbes, microbial isolates. All right, they are very important because they grow bacteria that don't require oxygen, hence anaerobic, the term. All right, and hence we're able to grow them well on this reducing media. Right, another type of media is called assay media. Right, assay media itself, it's used by many of the scientists to test effectiveness of antimicrobial drugs. Okay, and as well as drug manufacturer to assess the effect of disinfectants. All right, so most commonly they're used to test effectiveness of antimicrobial drugs. All right, you have carbohydrate fermentation media. Carbohydrate fermentation media, they contain sugars that can be fermented. So once sugars are being fermented, acids are being produced, and a pH indicator will be used to show this reaction. So it helps to identify microbial sugar fermenters. All right, we also have enumeration media. Enumeration media are often used in the industry and in environmental laboratories, right, to count or to enumerate the number of microbes, to quantify them in samples such as in your food, your milk, and the water, or in the soil, or other samples. And finally, we have the enriched media, and this enriched media allows fastidious microbes to grow. Like for example, we saw earlier the blood agar, okay, as well as other media such as the chocolate agar. Okay, apart from all the culture media that we have seen, they're mainly used for microbes such as bacteria, fungi, right? There are also culture media for viruses as well. So remember from earlier topics, right? Viruses are oblique intracellular parasites. They must be provided with a host living cell to replicate. Okay, so some of these culture media includes the bacterial cell culture, okay, the embryonic eggs, the living animals, and plants, as well as tissue and cell cultures here. So I want to switch gear for a bit when we bring our focus over to the preparation of microbiological media. So we have talked about the different culture media itself, but how is it being prepared? Often the culture media themselves are supplied as lyophilized powder and are stored in room temperature, or they may be refrigerated. So often we will reconstitute them with DI water and then autoclave it before use. So in the practical, you will be having the opportunity to prepare this media, such as the nutrient agar, right? While you are preparing it, it's important to adhere to aseptic techniques, because we want to minimize the contamination of the culture media intended for culturing the microbes of interest. So I have included some YouTube videos here to show you how we actually can prepare nutrient agar, as well as how to pour agar plates using aseptic techniques. So have a look. So I also want to mention about additional supplements. So sometimes we do add additional supplements such as growth factors or antibiotics into our culture media here. So often we can bought them commercially as lyophilized powder or stock solution. And these are usually kept refrigerated or below 0 degrees C, freezing point. So if some of these supplements are sterile, like for example antibiotics, certain antibiotics are provided sterile. If these are sterile powders and the powders are eventually reconstituted with sterile water, then these sterile liquid supplements can be added directly to the sterile media when they are cool. And for agar, when it's still molten, it's around 45 to 50 degrees. In contrast, if the supplements are reconstituted, all right, but they are non-sterile preparations, and then you will need to add them to a sterile media itself, right? Then what you can do is you can actually filter sterilize these supplements first before adding, because after addition to a sterile media itself, and often these are in large volumes, it doesn't make sense to sterilize the whole large volume of media itself. So in this case here, if it's a non-sterile preparation, those reconstituted non-sterile preparation supplements can be filter sterilized, typically through a membrane filtration before use. So the sterile liquid media prepared may be refrigerated in the fridge for up to several weeks. Solid media itself, like agar, has a shorter shelf life relative to the fact that because they are subjected to dehydration as well as contamination. The media may contain certain labile components such as antibiotics and growth factor, which will eventually lead to a shorter shelf life for the media itself. So whenever in doubt for storage and preparation of the culture media, we will always refer to the manufacturer's instructions. Usually this comes in the form of a brochure or leaflet to inform the user on how to prepare the media as well as how to store it. So I want to talk about the different various methods to maintain or to preserve microbes. Depending on the type of microbes and the required duration that you need, you can preserve them for short term to long term. Let's look at the short term first. Short term, we are looking at days to month, and often we will refrigerate them so that we can slow down the microorganism metabolism. You will often see that we will actually keep the petri dishes with the agar plate, all right, both inoculated in the fridge so that we can actually keep them for longer for future use, but not too long, about a few months only. Subculture is also possible. We talk about subculture in earlier slides. So by doing subculture, especially if it's liquid broth or liquid media, all right, we can actually subculture the microbes into fresh media to keep them going. Okay, the third point here will be using a slant culture or step slant culture. The step slant culture will be similar to a triple sugar iron like this. So in this case here, you directly inoculate into the solid agar in the slant tubes. Such as TSI agar. The main purpose is probably not to look into the differentiation part in how the different sugars have been fermented to identify microbes or even even identify any gas-producing microbes. But importantly, the inoculation of them into the solid agar and slant tubes like that actually give you very good short-term preservation method to retrieve microbial culture with sufficient quantities. For long-term preservation methods, and we're looking at years here, so often we will actually freeze down the microbial culture with glycerol. Bacteria are typically frozen in glycerol and then stored in the vapor phase of liquid nitrogen. So glycerol in this case is a cryoprotectant that helps to prevent the microbial cells from bursting due to the crystals formed from freezing. All right. Another aspect we can do for long-term preservations is to do freeze drying, also known as lyophilization. So the culture is frozen, and then we can dehydrate them by sublimation under the vacuum. This will change the culture into a dry, stable powder that we can even store at room temperature or at a fridge, allowing us to keep for years to come, in case we need it in the future for either experiments or other culture use. So for today, as summary, we have talked about the different types of microbiology media based on the function itself. So the general media is a all-purpose media itself. You can allow you to grow all types of microbes. All right. Some example of this are your LB agar or even your nutrient agar. All right. Your minimal media often contains the media with a minimum number of substrates that will allow you to identify which of the microbes are able to survive on these substances or constituents. One example of this is the M9 salt agar. You have selective media as well, right? Selective media, the name suggests, allows you to select for one microbes, all right, and from the others. So it favors one microorganism growth, but it may inhibit another growth of another microorganisms. All right, so a good example of your selective media is your mannitol salt agar. Okay, we have also talked about differential media. All right, differential media utilizes color changes in terms of a pH indicator to determine, for example, if there is acid or alkaline production by the microbial cultured on the differential media. So the utilization of the nutrients in this case, it could be a carbon source such as your lactose, sucrose, or glucose. Right, different microbes utilizes different carbohydrates and hence lead to a different color change due to the production of either of acid or alkaline, okay, changing the pH of the media. One good example of this is your McConkey agar. All right. We also have enrichment media. Some of the enrichment media well known is like blood agar. Right. The blood agar contains blood, okay, typically from sheep, and they typically allow the growth of fastidious microbes that are very fussy. They need blood in order to grow well. If not, they can't. Right. There are also specialized media. Some of these specialized media are used in industry, such as the assay media, as well as the transport media, so that you can utilize them for studies, assays, to look at whether they are responsive to certain drugs, antimicrobial drugs specifically, as well as keeping them stable between probably one lab to another when you need to transport some of the cultures between laboratories. All right. We also look into the use of appropriate media for various purposes. So some of this microbial media itself, they are often used for growing, isolating, differentiation of the different microbes. All right, and how are they used in the different scenarios here? We have also talked about. Okay, and finally we talk about the different methods for media preparation. Okay, the culture media itself are typically prepared under aseptic techniques, and if you have certain constituents that are sterile or non-sterile, you need to make sure that they are filter sterilized first to render them sterile before adding it to the sterile media, because we want to just culture the intended microbes here. All right, and then finally we talk about the different ways of short-term and long-term preservation of microbes. Whether is it for short-term, you can keep it in the fridge okay, or for long term you are freezing it down with glycerol. Okay, so I've included some recommended reading here. I will highly recommend that you read through the chapter 3 as well as chapter 9 in the recommended text of Cohen and Smith, Microbiology, a System Approach for the 2025 edition. Okay, this is available in your Brightspace LMS Connect platform. So do take time to read up these chapters. I believe that this will greatly enhance your understanding in this topic. All right, in terms of the culture method itself, okay, and also the manipulation of microbes, you can find more information on chapter 3 and chapter 5 here in the text by Benson's microbiological applications. This is available in the library as well. So that's all for topic 3, right? So I will see you in topic 4. Thank you very much.